Cool. Hello everyone, my name is Dagny. I'm a PhD student in the Department of Natural Product Biosynthesis in Professor Sarah O'Connor's lab. And today I will talk to you about how to manipulate the metabolism in 3 difficult steps. So first we should consider what metabolism actually is. And metabolism is defined as the chemical processes that occur within a living organism in order to maintain life. And the way we like to think about metabolism as humans is mostly eating food. So we get our nutrients from different food sources, we digest them, we can run around happily and go about our business. And this is the basic view that we have on metabolism, but actually metabolism is way more complex and it looks a bit more like this. So this is a map of metabolic pathways in living organisms that you can find online, and I actually highly recommend it. It's from this database called KEGG. And in here you can see that there are a lot of different branches of metabolism that are present in living organisms. And you see that basically metabolism is a very complicated network. And this is what we as scientists are trying to untangle. So we are trying to get to know how each one of the branches works. And today I'm going to talk to you about a little bit how we can manipulate those metabolic processes, how we go about this. So to make this a bit simpler, I would like us to split metabolism into 2 categories. The first category is primary metabolism. And basically, primary metabolism is the production of essential building blocks. So for living organisms, the essential building blocks are sugars, amino acids, nucleotides, lipids, and energy sources. And I like to think of primary metabolism as, I like to imagine it as 2 things. The first one being a gas station, so something that you get your fuel from, and you have your fuel, you can use it, you can do whatever you want to do because you have the energy to do it. And then the second thing is bricks. So as I said, primary metabolism produces essential building blocks, and these are simple building blocks, just like bricks. You can build different things from them, but you need the primary metabolism to provide the building materials. And today I am not really going to focus on primary metabolism and the manipulation of primary metabolism, but I just wanted to give you a quick example of the manipulation of primary metabolism. And please do not be scared of this scheme. It looks a bit complicated, but I just wanted to focus on 2 of my, I think, favorite examples of engineering of primary metabolism. So it's engineering of the production of biofuels from primary metabolism. And here you can see there are 2 examples of organisms that were engineered to make those biofuels. The first organism being Escherichia coli, which is this organism right here, and it was engineered to produce long-chain alkenes, which can serve as fuels. And then the other organism being Saccharomyces cerevisiae, which is basically baker's yeast. This is the same organism that we use to brew beer. This is the same organism that we use to make bread. And this can also be used to produce some useful biofuels. So when you look at this scheme, you can see that we start from glucose, from this basic molecule that is then turned over into different products by a series of enzymatic steps. And these enzymatic steps that are highlighted in red and green are actually primary metabolism steps. And in here you can see that these pathways have been engineered by addition of some extra enzymes to produce some useful chemicals. For example, farnesene, as I said in Saccharomyces cerevisiae, or some other biofuels that are quite efficient, and they are pretty good for use because they have less environmental impact than traditional petrol fuels, as they can be produced from waste that comes from other industries such as farming. So this is just one example of primary metabolism engineering. But today I would like to focus mainly on the engineering of the secondary metabolism. And what is secondary metabolism? Well, secondary metabolism consists of metabolic pathways and products of metabolism that are not absolutely required for the survival of the plant/organism. I am saying plant/organism, plants are organisms, but today I will focus mainly on plant secondary metabolism. And curiously, secondary metabolism is responsible for many bioactive compounds that are used medicinally. And so these two photos that you see on this slide, this is a continuation from my primary metabolism slide. So as I said, primary metabolism, I like to think of it as a gas station and a pile of bricks. Well, for me, secondary metabolism is like a nice car. So you have your fuel and you have this tool that you can do cool stuff with. So you can go for a nice road trip, you can go get your groceries, and it basically can make your life easier and much cooler. And then the other picture is showing a kitchen. So I like to think of secondary metabolism as a kitchen because with just bricks, products of primary metabolism, you can get a basic shelter. But if you have a nice kitchen, oh boy, we can invite people over, we can cook nice meals, and it gives us a lot of opportunities to kind of broaden the scope of what we can do in our shelter. So this is how I really like to think of secondary metabolism. And then I put this figure here, this scheme, which looks also a little bit complicated. But this is just to illustrate that secondary metabolism is actually tied to primary metabolism. And this bond is very important. So in the small letters, you can see the products of primary metabolism, and in capital letters, you can see classes of different secondary metabolites. And as you can see in this metabolic network, basically all the secondary metabolites come from the products of primary metabolism, which is a very important thing to remember and consider when engineering secondary metabolism in living organisms. Okay, so I said that we are going to focus on plant secondary metabolites, and indeed, here are some examples of plant secondary metabolites. So the first one is caffeine, which is, it's got quite a simple structure in comparison to the other ones, and it's a metabolite that we all know and love. We basically use it every day. I use it every day at least, to keep ourselves awake. And this is produced by different plants, the coffee plant, the tea plant. And actually humans use it to stay awake, but this is not what plants use it for. In plants it has a different function, mainly in defense of these plants. And then we have some other examples, such as, well, let's go to Taxol, which is anti-cancer compound from the yew tree. And you can see this is quite a complicated scaffold. It is actually used in chemotherapy, and yeah, it's a very potent chemotherapy agent. Then we have artemisinin, this compound right here, that comes from this plant Artemisia annua. And I put this here because I think this is the most well-known example of engineering of plant secondary metabolism. So basically, artemisinin is this compound that has anti-malarial properties, and its production has been engineered in the organisms Escherichia coli, a bacterium, and Saccharomyces cerevisiae, which is the baker's yeast that I was talking about earlier. And I think this is one of the first examples of plant secondary metabolite engineering, and probably one of the most well-known ones. And then we have this molecule right here, which is called Vimblastin, and this is the favorite molecule of people in my lab. So this molecule comes from the plant Catharanthus roseus, the Madagascar periwinkle. And you can see it's got quite a complicated and interesting structure. Actually, it is also used as a chemotherapy agent. And it's of big interest to the pharmaceutical industry. And then the last compound right here is noscapine, which comes from poppy plants. And noscapine is actually the compound that I am going to focus on in the further part of my talk. So this is kind of a case study we're going to use to illustrate how the manipulation of plant secondary metabolism is done. Okay, so I showed you before the complicated compound Vimblastin, which right now on this slide you can see right here. And I wanted to put the pathway, biosynthetic pathway leading to production of Vimblastin in the slides because I think this is a very good illustration of how complicated the biosynthetic pathway is, and the really interesting chemistry that is going on in the plant to produce this secondary metabolite. So in here you can see that it actually starts from a primary metabolism product, and then there is a series of steps, a very long series of steps that leads to the production of Vimblastin in the end. And each one of these red words is name of an enzyme that catalyzes the formation of next biosynthetic intermediate on the way to the production of Vimblastin. And the discovery of this biosynthetic pathway has actually taken quite a long time and a lot of effort by a number of different labs all around the world. And it has been elucidated. In 2018, we published a paper with the last steps of the production of Vimblastin. And so now this can also potentially be a target for some metabolic engineering efforts. But yeah, the main highlight of this slide is that it's very complicated. Secondary metabolism is quite a complicated thing. And so the complex nature of secondary metabolism is one product is one problem on the production of these medicinal products. There is another obstacle, which is that the plants actually produce very tiny amounts of these compounds. So you can have a truckload of leaves that you can extract chemicals from, but you will still get probably very small amounts of these bioactive compounds. And this is one of the main challenges that people that engineer metabolic pathways are facing. So we want to reconstitute the chemistry that is done by the plant in a more suitable host that will allow us to produce the chemical of our choice. And also we want to increase the yield of the production of such chemicals so that this production platform is actually relevant for the industry. Okay, and now I just wanted to introduce to you three main areas of research that focus on providing tools for the manipulation of metabolism. So starting with the first one, biotechnology, and I think this is the oldest one, I would say. Probably. And biotechnology is defined as the exploitation of biological processes for industrial and other purposes. And I remember when I was first learning about biotechnology, probably in 4th grade, we were learning about pickling, we were learning about brewing, generally stuff like that. So processes that actually exploit the organism's natural metabolism in order to provide some advantage for humans. This is why you have this photo of sauerkraut here. So this is a very good example of primitive use of biotechnology. Then we have this other area called metabolic engineering, and I think this is a bit of a hot word. So this area has emerged in late 80s, early 90s. And it is defined as the improvement of product formation of cellular properties through the modification of specific biochemical reactions or the introduction of new genes with the use of recombinant DNA technology. And the use of recombinant DNA technology is very, very important in this context. It enabled a lot of different investigations into metabolism. And I think this is the area of research that is mostly used to manipulate the metabolism, as the name suggests. And then we have the third and last area that I would like to talk about, and this is my personal favorite, and it has a very, very fancy name, synthetic biology. So synthetic biology is a field of biology that draws on principles of engineering to create new biological parts, devices, or systems, and to redesign or improve natural systems. So basically, there is a huge debate about the definition of what synthetic biology actually is. But for me, synthetic biology is this platform that we can use to improve living organisms or design living organisms de novo with properties that we would like them to have. And one of the examples that I think is super cool of the use of synthetic biology is the total synthesis of Escherichia coli with a recoded genome. So basically what the research group did in this investigation is they synthesized a bacterial genome, and they basically created a bacterium that has synthetic genetic information, and it is alive and it works, which for me is actually quite amazing. And then we have this other example, and this is more of a visual example. So this is the repressilator system, and this is, I think, the first synthetic gene circuit that was put together. And basically, just a quick explanation of what this figure is and what the repressilator actually is. So in here, as you see, we have this fragment of some genetic material that we put into bacteria, and it has 3 genes on it, TET-R, LAG-L, and CL. And then there is another fragment of genetic material that is also put into bacteria that is green fluorescent protein. So it's basically a protein that fluoresces in green. And what the system does is that these genes are actually, their expression is dependent on each other. So the expression of one gene actually inhibits the expression of the next gene. And so you can see that the expression is kind of like forming this wave pattern. And this is reported using the different fluorescent proteins that emit different colors. So I have this quick video here where you can see E. coli cells going through a narrow capillary, and they are changing color. And this is due to the fact that these genes are expressed, and the negative feedback loop is working. So the cells are actually changing color depending on which gene is currently the expressed one. And I think this is quite an awesome and very visual example of what we can do with synthetic biology tools. So yeah, that's synthetic biology. And then I just have a couple of examples of photos that I found on the internet while looking for graphics for this lecture. So when I was Googling biotechnology, synthetic biology, and metabolic engineering, I came across these beautiful pictures. And I must say that actually all of them have a grain of truth to them. So let's start with the one in the bottom left corner. You can see someone's just holding 2 bell peppers, and there's some pills just scattered around. And I think there is some logic behind that, because as I told you before, plants can produce secondary metabolites that can act as medicines for humans. So I think, although this is not the best representation of this process, I think this kind of makes sense. Then we have this photo right here, a sausage wrapped in a banana peel, and I rhymed, fantastic. And well, I wouldn't really do that. I don't know why anyone would wrap a sausage in a banana peel, but I think there is a thought behind it, and the thought is probably someone was thinking about the engineering of artificial meat, which is actually another hot topic that people are talking about quite a lot. So although bananas probably have nothing to do with artificial meat, I think there is some logic behind it. And then the photo in the bottom right corner, well, this photo is true because this is what I look like after 8 hours of reading biotechnology papers. And then the photo in the top left corner is actually surprisingly very accurate, because I even found a paper where scientists were trying to introduce some foreign gene material into tomato fruit using a syringe and a needle, which is really cool. The only thing that I think is wrong with this photo is that these gloves are not suitable for lab work. These are cleaning gloves, so I don't think we would use that in the lab. But apart from that, this is quite a good representation of biotechnology. Okay, so back to our manipulation of metabolism. I just wanted to give you a brief case study of how this process is actually working in practice. So the title of my talk is Manipulate the Metabolism in 3 Difficult Steps. And those steps are design, build, test. This is called the DBT cycle. And this is the cycle that we use to engineer biosynthetic pathways in living organisms. And the pathway that I would like to focus on is the noscapine pathway in the poppy plant that I mentioned before. So going through the steps of the cycle, first we have the design step. And well, to design the reconstitution of a biosynthetic pathway, first we have to know what the pathway actually looks like. So we have to do some pathway discovery. And this step is actually, well, on this scheme is quite a small step, but it's a huge effort that scientists put into the work to actually find out what those biosynthetic pathways look like, what are the steps, what are the enzymes that catalyze the reactions of these pathways. So this actually takes quite a long time to find out. But once we have the discovered pathway, we can design the reconstitution of this pathway in a suitable host. So there are many different molecular biology techniques to deliver foreign genes into our host organism of choice. I am not really going to get into this, but there is a lot of different platforms that we can use to do that. And actually the design of the reconstitution of biosynthetic pathways, it's a very important aspect of this cycle, because it basically determines what yields we're going to get from our reconstitution, if it's going to work or not, if we can channel the substrate through the biosynthetic pathway. So this is a very crucial step. And there are actually a lot of bioinformatics tools that are available for scientists to use when designing the reconstitution of biosynthetic pathways. So this is something, an area that tools are still being developed for, and it's actually quite rapidly growing. And then now we have the design, then we can go to the build step. So in here, I have this figure from this Lee et al. Paper from 2018 with the design of the reconstitution of noscapine in Saccharomyces cerevisiae, which is this organism that you can see growing here on the plate, the baker's yeast. And basically, what we do is we introduce those foreign genes from, in this case, from the poppy plant, into our organism, our host organism of choice. And that way we build a strain that we think is capable of producing the metabolite that we want. And then the last step of this cycle is test. So actually this is the most exciting step, I think. We basically take our organism that we modified, and we can culture it. This is a photo of a fermenter and this is quite actually a small fermenter. I've seen way bigger fermenters. This is, I think, a pilot plant fermenter that you would use to grow your engineered organisms. And So you put your yeast cells in there, you culture them, and then after a certain amount of time, you can take out the metabolites that were produced by these yeast cells. Either they are released into the medium in which the yeast cells are cultured, or perhaps you have to destroy the yeast cells and actually just force those metabolites out of the dead cell yeast bodies. But the purpose of this is to have some extracts that you can then analyze using analytical chemistry techniques. And then when you have your results, there are two things you can do. If you are not happy with your results, then you go back to design and you repeat the whole cycle and see if you can improve your design, see if you can improve your yields. And if you are actually happy with what you get from this analysis, you have your engineered production host and you are happy with the yield of the product, you are happy with the purity of the product, and you have this strain ready to use and perhaps to actually be used by some industrial partners for the production of the desired metabolites. And in here I just have this short citation from the Lee et al. 2018 paper. I just put it here because I think it's quite impressive what they achieved. Optimization directed to tuning expression of pathway enzymes, host endogenous metabolic pathways and fermentation conditions led to an over 18,000-fold improvement from initial noscapine titers, and the final noscapine titers that they obtained were 2.2 milligrams per liter. So 18,000-fold improvement, I think, is very impressive. I think 2.2 mg per liter, it probably still has to be improved a little bit for it to be actually adopted in the industry, but I think this was a monumental effort, and I think overall this paper and this investigation is very, very interesting and a very good example of the manipulation of metabolism. And so this is actually my last slide, and I just wanted to tell you a little bit about what the Miami consortium that I am also a part of and who is the partner of this series of talks, what we actually do as this consortium. So basically we are also focused on plant secondary metabolites, and we're mainly focused on these two plants, Catharanthus roseus and Rauwolfia tetraphylla. And I showed you before some examples of secondary metabolites from these plants that have interesting bioactivities. And as the Miami Consortium, what we are trying to do is actually discover these biosynthetic pathways that lead to production of these interesting compounds. In our case, it's monoterpene indole alkaloids. This is where the first part of our name actually comes from, MIA. We are trying to make the production of these monoterpene indole alkaloids more accessible in microbial platforms. So basically, we're trying to get the biosynthetic pathways to go from the plants into the bioreactors and then possibly further to the industry, so that we can use some useful we can get some useful compounds in good yields, and perhaps maybe lower the price of these useful compounds for the people and the institutions that need them. And with that, I would like to finish my talk, and I am very happy to take any questions if there are any. Thank you very much.
Yeah, Ola Julia asks, why do you like synthetic biology so much?
Well, there is a lot of reasons. The two reasons that I put in there, I think those are like the most visual reasons that I always give. But what I really like is, I think the philosophy behind it is that we can not only use the traits that the organisms already have in them for human advantage, but we can also use biological systems to produce new tools that can help us engineer organisms to for human advantage. And I think this is an awesome part of synthetic biology. And one other reason is, and this is more of a practical reason, is that synthetic biology is, I would say, quite standardized, and there is a lot of sharing in synthetic biology. So there is a lot of common genetic parts that people use and share in between labs, and I feel like the sense of community within the synthetic biology area is also quite a nice thing, and one of the reasons why I like it so much.
Okay. Say goodbye.
Okay, cool. Then thank you very much for tuning in.